What if we could cure diseases by targeting the root cause, our DNA? Nucleic acid based therapies are transforming medicine, and gene therapy for Duchenne muscular dystrophy is one of the most exciting breakthroughs. This genetic disease without a cure has affected thousands of families, maybe even someone you know, but scientists are developing treatments to fix faulty genes and stop DMD. In this video, we'll explore how gene therapy works and its transformative impact on those living with DMD. Welcome to the genetic basis of stuff and things. What is nucleic acid based therapy, and how is it helping us fight various debilitating diseases? Nucleic acid based therapies involve the use of DNA, RNA, or related molecules to treat genetic or molecular diseases. Unlike novel treatments, which target the cause of the disease, nucleic acid based therapy targets the source directly, the mutated protein or gene, which can yield long lasting or even curative effects through gene inhibition, addition, replacement, or editing. Some examples of nucleic acid based therapies include mRNA immunotherapy, CRISPR Cas9 gene editing, antisense oligonucleotides, and adeno-associated virus vectors. One of the most exciting advances in nucleic acid-based therapeutics is gene therapy, which involves the delivery of corrected copies of genes into cells to treat or prevent genetic disorders. An example of this is using adeno-associated virus vectors to carry and insert these transgenes into the nucleus of the patient's cells. Recombinant AAV vectors are excellent delivery vehicles as binding interactions can be modified, enabling specificity and variety, whilst also displaying low immunogenicity, allowing for lower risk of adverse effects in patients and higher efficacy. But what role does adeno-associated virus vectors play in treating diseases? The answer lies in revolutionary developments in treating Duchenne muscular dystrophy. Duchenne muscular dystrophy is an X linked recessive progressive muscle wasting disease affecting 1 in 5,000 male births. The disease is caused by mutations in the DMD gene, which codes for the dystrophin protein. Dystrophin localizes to the sarcolemma of skeletal muscle, forming a major component of the dystrophin associated protein complex, which acts as a mechanical link between the cytoskeleton and extracellular matrix. This complex is essential for optimal skeletal muscle function. Frameshift mutations, usually deletions, insertions, or duplications, result in a prematurely truncated DMD gene, resulting in structurally and/or functionally unstable dystrophin proteins. Mutations occur most commonly in exons 45 to 55 and 3 to 9 in approximately 47 and 7% of patients with DMD, respectively. Boys with DMD typically show symptoms between ages 3 and 5, including gross motor delay, gait issues, and difficulty standing. As the disease progresses, it can lead to cardiac and respiratory failure. Current treatments like corticosteroids aim to reduce inflammation and slow the decline in mobility and respiratory function. In 2020, Dr. Jerry Mendell and his team developed an AAV vector drug that successfully increased dystrophin expression in a phase 1/2A trial. Since the full DMD gene is too large for the AAV capsid, they designed a micro-dystrophin transgene with key components of the DMD gene to produce functional dystrophin. Packaged in an AAV RH74 capsid, the transgene includes an N terminus for binding to F actin, spectrin repeats 1 to 3, spectrin repeat 24, and a cysteine rich domain. Spectrin repeats 1 to 3 directly attached to the sarcolemma, reducing muscle injury. Spectrin repeat 24 is crucial for microtubule binding, while the cysteine rich domain is vital for binding to the beta dystroglycan. These components work together to promote the reconstruction and restoration of the dystrophin associated protein complex. The microdystrophin was placed under the control of the MHCK7 promoter, known for its high expression levels in skeletal muscles, compensating for the inability to fit the entire DMD gene into the AAV capsid. Results showed 81.2% of muscle fibers expressing microdystrophin, with 96% intensity at the sarcolemma level. The patients demonstrated improved mobility and muscle growth post treatment. It is evident that microdystrophin transgene therapy yields greater functional improvements than corticosteroid treatments, pointing to a future where DMD could be treated at its genetic source rather than merely managing symptoms. As research advances, gene therapy for Duchenne muscular dystrophy shows great promise. Ongoing clinical trials focus on refining these treatments to improve their safety and effectiveness for a broader range of patients. Following the success of the clinical trials in 2020, Mendell is leading a larger scale phase 2 trial expected to conclude in 2026. Meanwhile, Pfizer is conducting a phase 3 clinical trial with 122 patients, testing the clinical efficacy of a similar AAV vector based micro-dystrophin therapeutic. Progress being made today could pave the way for treating genetic diseases like DMD at their source. Gene therapy is opening doors that were once thought impossible, and every day we're getting closer to breakthroughs that could change lives. For those affected by Duchenne muscular dystrophy, either personally or within your family, advancements in gene therapy offer a new level of hope. Viable treatments are closer to public access than we think. The future of medicine isn't far off. It's here, and together we are shaping it.